Hello everyone, welcome to today's live broadcast, Genomics in Public Health Microbiology. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Professor Ben Howden, PhD. Dr. Howden is an infectious diseases physician, medical microbiologist, and head of the Howden Laboratory located in the Department of Microbiology and Immunology at the Doherty Institute for Infection and Immunity. The laboratory is closely affiliated with the Microbiological Diagnostic Unit Public Health Laboratory, MDU PHL, where he serves as director. The Howden Laboratory works in close collaboration with the Listeria Laboratory and has close links with the infectious diseases microbiology units at Austin Hospital. The scientists' research uses genomics, molecular biology, epidemiology, and clinical studies to address a broad range of issues related to invasive bacterial diseases in humans, especially those caused by staphylococci, enterococci, and other antimicrobial resistant species. Working closely with scientists in the MDU PHL, Dr. Howden's group investigates the evolution, epidemiology, and spread of bacterial pathogens of public health significance, such as Neisseria gonorrhea, Listeria, monocytogenes, Shigella and Salmonella species, and Legionella species. I will now turn it over to Dr. Howden for his presentation.
Okay, thank you very much, Judy, and thank you for the opportunity to speak to you all. The topic I'm talking about is genomics in public health microbiology. And the reason I have an interest in this is through our research, I've used genomics for many years to understand how bacterial pathogens evolve and spread. And now, as a director of a public health laboratory, we're trying to apply this technology in real-world public health microbiology. If you think about what the game changers have been in public health or clinical microbiology over the last couple of decades, there's really two main ones. That of the development of the PCR reaction, which was in the 1980s and 1990s, and also the use of typing methods such as pulse fill gel electrophoresis, and more recently the development of multi-TOF related diagnostics. And these technologies have really changed the way we do clinical microbiology and also public health microbiology. And there's really no doubt that genomics is another one of these game changers. And we're just at the start of this evolution of genomics and how we're going to use it to really change the way we do public health microbiology. And why are we so interested in this technology? Well, as I've said, it's a disruptive or game-changing technology which can really transform the way we perform these practices. The era of genomic medicine is already here, with the technology being used in the diagnosis and treatment of certain genetic and disorders and cancers. But the application in public health and clinical microbiology is lagged behind a little bit. But there's really a major push internationally to catch up here and use this technology to enhance the practices of these microbiology specialties. So what are the potential applications then of genomics in these spaces? Well, they're very broad actually, and everything you think of, you can find a way to apply genomics to the question or the diagnostic assay or the typing assay that you're trying to do. But just as a brief summary, some of the key areas where genomics will have an impact is in the diagnosis of infectious diseases, the characterisation of antibiotic resistance mechanisms, but also mapping the way resistant bacteria spread, detecting and responding to outbreaks of disease, whether they be foodborne or other types of microbial diseases, tracking the source of outbreaks. This is particularly the case, for example, for foodborne disease, which I'll talk about a little bit more, where we might be able to use genomics to really pinpoint the source of contamination problems that result in outbreaks of human disease. And then there's some other applications such as characterization of unusual microbes, so looking at microbial species which are not typical pathogens in humans and characterizing them in more detail. And certainly also in infection control in hospitals, it has a similar type of approach to using it for foodborne disease characterization and detection, but using it in the hospital environment to understand how pathogens are spreading within hospitals and trying to disrupt this spread. And genomics will help in all of these areas. So why are we so excited about genomics in terms of the workflow in the laboratory? Well, here's just a slide that summarizes the sample workflow that might be how we work in the public health environment. So we get a lot of different types of specimens in the laboratory, whether they be from humans, food, environment, or animals. And we have a very broad range of diagnostic techniques and also typing techniques that we apply to these pathogens, and it means that we're doing a huge number of different assays within the laboratory, and this would be well known to anyone who works in a clinical or public health laboratory. And this leads to some reports that either describe the characteristics of the pathogen or possibly the characteristics of a group of pathogens that might determine whether they're related or not, and whether there's been some sort of spread of a particular pathogen. What are the limitations of this current workflow? Well, they've worked very well for us for many years and they've led to a lot of good microbiological outcomes, but they are reasonably low resolution, a number of these. And so we have a rough idea of what might be going on with the spread of bacteria, for example, but we don't necessarily have the detail. Some of them are labour intensive and some of them are relatively expensive in terms of time, particularly staff time and consumables. And so what are the potential advantages of a genomics workflow then? Well, it's the higher resolution, so this is the highest resolution we can really get in characterizing a microbial pathogen down to the nucleotide level. It's a single assay, so actually what we potentially can do is do a lot of our public health microbiology using a single type of assay and feeding in different microbiological questions into that. And this might provide data on strain type, resistance, virulence, and mobile elements. And it can also be very high throughput, so hundreds of samples can be multiplexed in an assay, and you can get the answer reasonably quickly. It's not within hours, but it's within days. And actually, it's relatively cheap when you're talking about a number of different assays that you might be able to replace with a single assay. Is it backwards compatible? We often get asked questions from the people we report our typing data to, can you tell us what the old typing results were as well? In some cases, yes. In other cases, no. And I guess there's going to have to be a transition to this new type of typing terminology and reporting, but there is certainly the possibility of some backwards compatibility, which I'll talk about a bit more in a moment. So what do I mean by a genomics workflow then? Well, this is just a summary of how it might work within the laboratory. So DNA is extracted from a bacterial species, for example. It goes onto the sequencer, and then a large number of sequencing reads are generated. And then the key next step is the bioinformatics black box, which I've got in the middle of the slide there. And this is where all the important analysis happens and where the meaningful data is generated from all of these sequencing reads that we get out of the genome sequencer. And what sort of information might we be able to get out? I've just got some examples there on the right-hand side of the slide. You could determine the resistome of the species, that is, does it have a particular type of resistance gene, such as MEK-A that you might find in Staph aureus, MRSA? Does it have a mutation in a particular gene, such as the C mutation I've got there, which might lead to a certain type of resistance, such as quinolone resistance in a number of different bacteria? You can also determine the relatedness of strains. I've talked a bit about this already, but what I've got there is an example of a phylogeny where you're looking at the distance between different microbial species and understanding how closely related they are to each other. And then some other things which are not necessarily directly related to public health service, but are also potentially interesting are virulence, gene characterization, and then also some more detailed analyses to understand evolutions of microbial populations, to understand how they're spreading and evolving over time. So just as an example from our own point of view, how do we set this up in a public health laboratory? Well, it's not straightforward actually, and so it's required a lot of investment in in hardware, and this is robotics and automated DNA extraction and various library QC modules that are required before you can place your sample onto the genome sequencer. And so these are the sort of things that might be required for a laboratory that is considering transitioning to a sequencing-based approach. You can outsource these tasks, but it does actually potentially increase the turnaround time. And if you're a laboratory that needs to have answers in a reasonably short turnaround time, then at this stage it's more likely that you're going to have an in-house approach. And then the key thing here, which I've highlighted, is the need for bioinformatic support. And this is Torsten, who's our main bioinformatician in our laboratory. But there's no doubt that you can't get the significant benefits of genomics without the high-level bioinformatic expertise that's required to extract the meaningful information from the data. So that's a key part of the setup. So before I talk about what we've been doing in our laboratory, I thought I'd discuss some of the international progress, because it's been very significant actually. There's been a lot of progress made in the application of genomics in public health and clinical micro, and these are just some examples here. So in the USA, the FDA has instituted a program of Salmonella and Listeria genome sequencing and using an online system called Genome Tracker, and I'll talk a bit more about that in a moment. In the United Kingdom, Public Health England has now transitioned to full genome-based typing of Salmonella species. So they get many thousands of Salmonellas referred to their laboratory each year, and they've now stopped the traditional typing methods, and every Salmonella species undergoes genome sequencing and rapid data analysis for reporting. And so their plan at Public Health England over the next few years is to transition to a total sequencing-based approach to public health microbiology. And there's also advances being made in Canada and other places. This is just an example of what is happening in the USA, and I think this is interesting to know because two things, mainly because it's the data is being made publicly available in a very rapid time frame, and they've generated a model where data can be shared across the country very rapidly to look for potential common links between human and food isolates of Salmonella and Listeria to try and define outbreaks and pick them up earlier. And so what's happening across the US is that a number of laboratories all have their in-house sequencer. Whenever they get a new isolate of Salmonella or Listeria, that isolate is sequenced, and then overnight that data is uploaded to something called Genome Tracker, which is housed at NCBI. And so every morning there's a new set of data in the system, and the FDA takes responsibility for reanalyzing all of that data overnight and then interpreting whether there's any new interesting relationships between isolates that should be investigated as a possible indication of a new foodborne disease outbreak. And just to give a sense of the volume of this sort of thing, this is up until December last year, the number of isolates of Salmonella and Listeria which are now in the Genome Tracker network, and it's well over 10,000 Salmonella isolates now and well over 2,500 Listeria isolates, just showing the volumes that are now being uploaded, and there's been this rapid increase over the last 18 months. So how does this help? I mean, we do have traditional typing methods, but how has this Genome Tracker approach helped? Well, these are slides that are provided by the FDA, just showing, I'll just point out here that this area here is the epidemic curve that you would normally see in a foodborne outbreak of disease, so that in the middle of this, through the epidemiological investigations, there would usually be recognition that there was an outbreak of foodborne disease, and then the investigations would start to try and determine the source of that, and using typing methods as well, trying to disrupt that outbreak. But what's happened here is this is an example case where earlier in the outbreak, before it was clinically obvious, the Genome Tracker system was able to pick up very close genetic relationships between bacteria that had been uploaded from the laboratories across the United States, and able to recognize that there was probably a contaminated food source. And that's led to an investigation that intervened and likely led to the prevention of a large number of foodborne disease outbreaks in this case. That shows the potential power of genomics if you have data sharing in real time to really disrupt these sort of things. So there's some examples of what's been happening internationally. just as an example of how we've tried to approach this locally for any laboratories that are considering a similar sort of path, what we've done is we've developed an applied genomics centre that sits within the public health laboratory, but is also within the research environment of the university where the laboratory is located. And what that means is that we have expertise both in public health microbiology, but also in the basic science of bacterial genomics. And so those two groups can work together to develop techniques that are going to allow for really applied genomics approaches to mapping diseases. And interestingly, once you start doing this, then the interest in the technology and the application of the technology starts to really become very broad. And so quite quickly, we've been asked to look at things like outbreaks of disease, such as VRE or vancomycin-resistant enterococci in hospitals. We've been applying it to Legionella when there's been potential outbreaks of Legionella, and also drug-resistant bacteria such as KPC, Klebsiella pneumoniae, which I'll talk a bit more about in a moment. There's also been a lot of work looking at foodborne disease, and as I've mentioned internationally, this is where a lot of the work has been done, and so we've followed a similar path to try and see whether we can get similar benefits from the technology that we've been seeing internationally. And then there's other applications, such as looking at tracking of antimicrobial resistant species, and also looking at other things such as sexually transmitted diseases. And so reasonably quickly you start to get a lot of potential investigations and requests to look at things, and it can actually generate a lot of work very quickly if you're not careful about how you strategically implement this. Okay, so some examples then of how the technology has been applied and how it appears to be helping in what we're doing from a public health laboratory point of view. So the first is an investigation of carbapenem-resistant Klebsiella pneumoniae that we've been undertaking in Australia over the last 12 months. Many of you who work in the clinical laboratory will know that carbapenem-resistant Enterobacteriaceae are really a very urgent international antimicrobial resistant health threat, and this is why, because essentially on the left there, they're resistant to almost all antibiotics. This particular outbreak, a number of them were susceptible to gentamicin but resistant to everything else. And there's a high risk of mortality and morbidity associated with these bacteria. If you look internationally, this is just a map showing in red areas of the world where there's been a largest number of cases of KPC, Klebsiella pneumoniae. You can see there are a number of regions where this has happened, but Australia has had very few cases to date. But what's happened over the last couple of years is that these are the isolates that have been referred to our laboratory for characterisation of CRA gene detection, and you can see that there's been increasing numbers of carbapenem-resistant Enterobacteriaceae, and in particular larger numbers of KPC2, Klebsiella pneumoniae. And so there was a particular type of bacteria also with the same resistance element, and this made us wonder whether in fact there was some local transmission going on, because previously most cases have been internationally imported. So how do we look at the isolates to try and determine this? Well, I guess traditionally we would have done something like look at the antibiogram, and this is the summary of the isolate antibiogram, and you can see that's actually a little bit heterogeneous, and so that wasn't particularly helpful. All isolates had the same resistance gene when it was sequenced, so it was all KPC2. And it was really impossible to tell from looking at them from this point of view whether they were related or not. Pulsatile gel electrophoresis could also be performed and did suggest that the isolates were quite clonal, but it's very hard to draw conclusive information from the PFG results. And so we undertook some genome sequencing of the bacteria, and this is just a slide summarising where the bacteria all fit. So all of the ones that we'd had recently fit within this part of the tree here. And so this is a phylogenetic tree where the horizontal distance describes the diversity of the bacterial species. And so these bacteria here, which have a long branch length, are very different to the other ones, whereas the ones that down the bottom here in red that have no horizontal difference in their then they're very closely related. And then the other thing we did was, because we're using genomics, we're able to compare our bacteria to the international publicly available data set. So over here on the right, in black, are all the international isolates of KPC, Klebsiella pneumoniae. And you can see they fall into two different clades when you compare them by genome sequencing. And then when you look at where all the ones from our outbreak have occurred, they're all clustered together and they appear separate from the ones that have been found internationally, or at least uploaded to the international databases. So if you zoom in then on this part of the tree where the very clonal looking strains occur, so this part of the tree here, then what you see is that you start to see some differences in the bacteria in this part of the tree, and it looks like there's 4 different groups that are 4 different clades of KPC2 Klebsiella pneumoniae circulating, and here they've been color coded by the referring laboratory. And you can see that there's a number of different laboratories with quite closely related strains. And down the bottom here you have a scale which is the number of mutations, but represented by this branch length. So that's 20 SNPs, so 20 single nucleotide polymorphisms, and you can see that a lot of the bacterial species have much less than 20 SNPs between them, suggesting they're very closely related. And using genomics, you can also get other interesting information, like defining the type of resistance gene that's present in the bacterial species, and in this case it was a particular type of well-characterized resistance plasmid. I won't talk any more about that. But the other thing you can do with genomics is also understand the pan-genome. So rather than just looking at the genome that's shared between all the species, you can look at the total gene content, and that's what this green image represents here. And so all of the bacteria, where there's green, they have the same gene present, and you can see there's very little variability between the bacteria in this tree, suggesting that they are very clonal or very closely related. So how do we determine whether there's transmission occurring across the whole tree, or whether it's part of the tree? Well, what we can do is look at the pairwise SNP distance. So that's how many mutations are between the different populations within that tree. And you can see here that there's a bimodal distribution, and so this left half here with less than 40 SNPs most likely represents groups of clonal dissemination. And so then if you apply that back onto the tree, what it means is that there's actually 4 groups which have been color-coded here that represent the 4 possible transmission networks, and I'm just highlighting them here. And so this just helps us try and understand the transmission dynamics. And so the top group here, they're all closely related, but they all come from different laboratories, and it's not clear whether there's been transmission. We need more epidemiological data, and that's our key message, is that the epidemiology is still very important. These next 2 groups here, the 2 groups are closely related, and they appear to have come from the same laboratory, so there's been probable transmission of wherever that laboratory is. And then further down you have isolates that are almost genetically identical, but they've come from different referring laboratories, and so could this be community transmission or common source exposure or inter-hospital transfer? And so the epidemiology is required to overlay onto this to really understand what's happening. And so we've now done the epidemiological investigation. It really confirms that the genomics has clearly described the population of these bacteria, and very clearly you can find close relationships between the patients that have similar isolates, even though they came from different referring laboratories. And so it's been an extremely useful tool to investigate this outbreak. And so the summary is that the genomics shows the majority of the recent bacteria that out in Melbourne are similarly closely related. They have a particular type of resistance gene, and there appears to be 4 transmission networks. And this has allowed the public health branch of the government to go and then investigate this further and implement some interventions to prevent further transmission. So that's an example of a drug-resistant bacteria. So now I'm just going to move on and talk about more of a foodborne disease, that of Listeria monocytogenes. It's a ubiquitous gram-positive organism. Human infections occur through foodborne transmission, and usually about there's 60 or 70 notified cases each year in Australia. And there's been a national surveillance initiative since 2010. So in the laboratories, the public health laboratories in Australia and internationally, there's a range of different typing tools that are used for Listeria. In some laboratories, up to 4 to 5 or 6 different typing methods are used, and this is just summarising what some of those typing methods are. It might be multi-locus sequence typing, PCR binary typing, multi-locus MLBA, and then PFG is the most definitive typing method. And so actually, when you think about it, that's a huge number of different methods and quite a lot of labour to perform those. And so what we've done here is we've applied genomics to our population, to investigate whether using a genomic approach will improve resolution, but also decrease workload for Listeria typing in the laboratory. And so here we have another tree, so a dendrogram describing a relationship between some Listeria isolates. You can see from the scale here that there's a very small number of SNPs on the tree, so it's only 25, so that all of these isolates here are essentially identical. And this is a group of isolates where the PFG, which is the highest resolution typing method here, is identical. And you can see that all the other typing methods on the right are essentially the same, particularly the PFG, the MLST, and the MLVA. So previously these isolates would have been considered the same, but when you look at the when you go back and look at the tree here, you can see that it's actually designated 4 different groups. And so what this has allowed us to do is have a higher resolution analysis of the genetic relationship between bacteria, and so that it's actually separated these into different groups. And when we go and check the epidemiology on this, it does actually validate the fact that these are different sources of contamination. And so the typing use of Listeria using whole genome sequencing really can replace a number of traditional methods. It has superior discrimination and is actually cheaper and more cost effective than using the traditional methods. We've also been able to develop tools where we can infer the MLST, the serotype and the binary type from the genome sequence. And so this allows us to report both on the genome sequence relationships, but also to report the traditional typing methods. However, it's not possible using short read sequencing to predict the MLVA or the PFG types accurately. So how do we report then? Well, this is a challenging part of transitioning to genomics, because we are reporting to epidemiologists who need to use this data to investigate potential common source outbreaks. And they're used to having binary type data, i.e. that is particular names or numbers given to certain typing results. So they might be used to a particular PFG group or a particular serotype, for example. And if you're reporting an analysis based on SNP distances between bacteria, it can be quite challenging for them to transition to this. And so this is, at the moment, the method we're using, where we're doing a phylogenetic relationship and looking at the SNP distances between bacteria, and then highlighting using a traffic light interpretation, i.e. red is highly likely to be related, green is not likely to be related, and yellow is in between, to really highlight the risk stratification of what cases should be investigated compared to others, and then the epidemiologist can apply that information to performing the epidemiological investigations. So that's all I was going to say about Listeria, but similar to Listeria, you could apply this sort of approach also to other bacterial species and other foodborne diseases such as Salmonella. I guess the problem with Salmonella is that the volume is much higher, so we're only getting tens or possibly hundreds of Listeria each year, but we get many thousands of Salmonella each year. So to make a transition from traditional methods to pure genomics for Salmonella is a lot of work and requires very efficient genomics workflows to make that possible. So I'm just going to talk a little bit more now about something that's a bit more in the research environment, but does significantly translate into understanding how we control drug-resistant bacteria in hospitals. And that's some work on the genomics of vancomycin-resistant enterococci. And so VRE are a common cause of colonization and infection in hospitals globally, and certainly have a very prominent issue within Australia. And we don't really understand why VRE remains such a problem. So nosocomial outbreaks of VRE are thought to occur by transmission between patients, and therefore effective infection control interventions really should limit cross-transmission. But despite implementation of such strategies, the incidence of VRE infections continues to rise in Australia. And we're particularly concerned because we have a very effective national hand hygiene program within Australia, and that's been very effective actually at reducing the number of MRSA infections. So here on this image you can see the number of MRSA infections at our particular hospital where we're investigating this. And back in 2001 it was very high, whereas the rate of VRE or VSC infection was quite low. And after the introduction of an effective hand hygiene program, we've seen a marked drop in MRSA infections. And then over that time, VRE infections have actually increased, such that in the late 2000s, for the first time, we had higher numbers of VRE bacteremia at our hospital than we did of the MRSA. And similar stories are being told around Australia, so it's not clear why that's occurring. And one of the interesting things about VRE in Australia is that we have a different type of resistance element. It's a VAN-B element, which is a bit different to what is common in the USA and in many parts of Europe. And the VAN-B element is interesting because many people actually carry this resistance element in their normal flora of the bowel, particularly within anaerobes within the bowel. And it's also been demonstrated within mice, at least, that you can take an anaerobe that carries this VAN-B element and mix it with a vancomycin susceptible enterococcus in the bowel, and it will generate a VRE. And so this made us think a bit more about what is actually going on with VRE in Australia and how common is this transmission of this resistance element between the anaerobes in the human gut and VSE isolates to generate new clones of VRE. And is this partly driving this issue of failure of infection control to stop VRE? And so this study here that I've put up the phylogeny from is looking at cases of VSE and VRE bacteremia at our hospital over a 10 to 12 year period and looking at the genetic relationship of the isolates. And so on the left here we have the phylogeny of the isolates, and so this is, as I said, over 10 to 12 years, and all the ones in black are the ones from our hospital, and the ones in yellow are ones from other hospitals throughout Australia and even an isolate from overseas. And the first thing you can see is there's a lot of diversity in this population, so that even at our one single hospital in Melbourne, we had isolates that represented those that were seen in other hospitals in Darwin and Perth in Western Australia, and also an isolate in the middle here that came from the USA. And so you can see there's a lot of diversity in the population that we're seeing at our hospital, and reflects the diversity that we see nationally and possibly even internationally. And then the other thing that we also note is that there's this real change in type of clone that we're seeing. So over the years, this type 17 VRE was seen throughout the whole time period, but in the later time periods we had the emergence of this new clone called ST203, which appeared to be associated with this rapid increase in VRE infections we were seeing at our hospital. And so what we did is we used the genome sequences to understand the resistance element. And so this slide here summarizes the characteristics of the transposon that carries the VAN-B resistance element. And we were looking at whether the sequence of the transposon was different. We also looked at the site that the transposon inserted into the genome of the VRE, the direction it was inserted, and also what the coupling sequence was around it. And this tree here is one that's just based on the transposon sequence, not on the whole bacterial genome, but just on the sequence of the transposon. I think one of the most interesting things here is that the isolates in red here are from anaerobes. So these aren't from human VREs, but they're from human anaerobes. And you can see that at least one of the anaerobic sequences of the transposon was totally identical to a large number of transposon sequences from human VRE isolates, suggesting that, or indicating at least, that these are essentially identical. So what we did then was we overlaid these characteristics of the transposon onto this core genome phylogeny that I showed you on a couple of slides ago. And so what we have here is all of the VSE and VRE isolates, and the ones that have a resistance transposon have these colours here, and ones that don't here are the VSEs. And so what you can see is that there's intermingling of the VREs and the VSEs. So here we have a VSE, and right next to it we have a VRE that's picked up a transposon. Here we have a number of VSE strains, and here we have a VRE that's picked up a transposon. And what we then did is we went through and looked carefully at the transposon characteristics and defined which of these strains or which of these isolates were new VRE clones, so that emerged independently. And each of these grey arrows here indicates a new VRE clone, and you can see from this that actually a lot of these VRE on this tree were actually new clones. So it It does not suggest horizontal transmission, but rather generation of new clones of VRE. However, this one here is an example of where all of the backbone core genome was the same and the transposon was the same, and they all came from the same time period, suggesting this was clonal dissemination, whereas a lot of the others were independent generation of VRE. And if you tally this up, what we had here was we had 36 VRE isolates in this population, and it appeared 18 of them were independently generated. And even for this new clone, so this new ST203, which is the one that emerged in the last 2 or 3 years, 9 of them appeared independently generated. And so this is very important for understanding the emergence and dissemination of VRE. And the conclusions and insights from this are that the vancomycin-susceptible E. faecium are a very important part of this problem. So it's not just about VRE and transmission of VRE, but it's the backbone, it's the VSE that really underpinned the problem of VRE in our hospitals, at least in Australia. And it's pretty clear from this that the gut anaerobes are potential sources of the resistance element, particularly this transposon 1549, and about half of the VRE likely arose by de novo evolution rather than transmission between patients in our hospital. So what then are the implications of these sort of genomic studies for controlling antibiotic-resistant bacteria? Well, a patient being treated with antibiotics could become a source of VRE, even if they're not exposed to another patient with VRE. So VRE screening and isolation will not recognise about the half of the patients that have this type of VSE. And identification and isolation of patients with strains of VSE, not just VRE itself, might be required to control VRE, or obviously that's a huge undertaking and may be totally unrealistic. It's probably going to be a combination of antibiotic control policies, enhanced cleaning, and universal precautions may be more effective for VRE. However, there was also clear demonstration of clonal transmission or dissemination on that previous slide, and so it's a combination of the two. It's not just about in vivo generation, but it's also not just about nosocomial transmission. And so this has really led to a rethinking of how VRE emerges and spreads in Australian hospitals, and what might be the best approach to try and control it in future. Okay, so I've talked mainly now to this point about how genomics will help us in public health microbiology, that is in foodborne disease, characterising isolates that are referred for molecular typing and looking at infection control interventions. But there's also a discussion at least about the role of genomics in a clinical microbiology lab. And if you think about this, then I think in future this will most likely be the case, but there's probably a lot further work to be done before it would be immediately transferable. If you think about how a clinical microbiology lab works, there's quite rapid processes we go through, from pathogen isolation to identification to susceptibility testing and reporting and clinical decision making. But if you look in the literature about the role of genomics in a clinical lab, you start to see a lot of papers emerging now where they describe different methodologies that might be relevant to the clinical lab. And that might be, for example, the direct detection of organisms from clinical samples. And so there's some papers now emerging on this. Identification of viral pathogens in clinical samples by real-time sequencing. And even quite a prominent case in the New England Journal of Medicine looking at the diagnosis of neuroleptospirosis by next-gen sequencing on a clinical sample. Obviously, the genomics workflow is not, doesn't easily fit within a clinical microlab at the moment, but there's also papers coming out describing methods for improving turnaround times and ways to approach how we might apply genomics in a clinical lab, such as sequencing directly from single colonies. And other papers that describe how this might be achievable. And then the other thing that we can do with genomics is we can rapidly look at antibiotic resistance elements, which I've already alluded to, and so there could be a role in future for predicting or characterising resistant isolates, although at the moment the phenotype is much quicker to perform than it is to perform a genome. One area where that may not be the case is in TB, for example, and there's quite a bit of work being done on using genomics to rapidly predict the resistome of Mycobacterium tuberculosis, either from broth cultures, or possibly even directly from clinical samples. And that is something that's probably most likely to come into practice earlier than other use in other areas of antimicrobial resistance. So if you look then at what the criteria that we use in a clinical laboratory to decide whether a test is useful or not, really genomics doesn't fit the bill just yet for a number of them. For example, in terms of the speed of testing, the genome sequencing really can't compete with the traditional methods, except possibly for tuberculosis. In terms of sensitivity, it doesn't really add much at the moment, although does it help with organism identification and susceptibility testing. In terms of specificity, yes, it's the highest resolution sort of characterisation we can get. Is it better than current alternatives? Not at the moment, although it might be complementary to some of them, particularly for tuberculosis or other unusual pathogens. And is it likely in future to alter patient management? In some cases, yes, for things where it's difficult to get answers with traditional methods, and then in future there might be more use of very specific genetic characterisation of microbial species to help inform treatment approaches, such as predicting novel mutation or finding novel mutations that might be associated with subtle resistance mechanisms, for example. So the current challenges in implementing next-gen sequencing in diagnostic labs are that the scale and capacity. So most genomics projects now are looking at hundreds or thousands of genomes, whereas in a routine diagnostic laboratory there might be hundreds of thousands per year, and so this is not achievable in the short term. There's going to be some difficulties in workflow integration, in particular with turnaround times. In terms of usability and expertise in the clinical environment, we don't have a lot of that at this stage. And then one thing we've found is there's even issues about data storage and security, and so each genome sequence we generate is a couple of gigabases of data, and so once you're starting to do hundreds or thousands, you run into problems with data storage and also data security, trying to share data between laboratories. And clearly the turnaround time to the final reports are relevant, and the limited expertise and cost. So just to summarise then, this is the summary slide for the talk. Really, genomics, technologies and bioinformatics tools will transform public health microbiology, there's no doubt about that. However, many existing methods in the diagnostic micro lab are unlikely to be immediately superseded by whole genome sequencing. It's partly due to a lack of knowledge and training amongst the clinical micro workforce, which is something we have to address. And the areas of most utility of diagnostic laboratories are likely to be resistance gene detection and epidemiological typing, so similar to the public health role, really. And in the short term, the expertise is likely to be concentrated in reference laboratories and tertiary centres. And really, in future, the outputs must be compatible with the requirements of a working laboratory if genomics is going to be introduced into the clinical microbiology laboratory. So just from a personal perspective, within Australia at least, what's the view to the future? Well, within public health microbiology, we would hope that all labs transition to a genomics workflow which is high throughput, fast and cheap. And most importantly, similar to the FDA model of a common online system to upload samples with some automated standard analyses and simple reports and visualization. And this could really enhance the practice of public health microbiology in Australia and enhance our ability to detect and respond to outbreaks of microbial disease. So that's my final slide, and I'm finished and ready for any questions and answers that you may have.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll answer as many questions as we can. So I'll give you a moment to formulate questions. If there are no questions today, I would once again like to thank Dr. Howden for his presentation. Do you have any final comments, Dr. Howden?
Thank you. Well, thank you for the opportunity to speak. I guess I would just like to say, it's a challenging area, and I think there's a lot of uncertainty about how we're going to move forward in this area over the next few years. But there's no doubt that the technologies are advancing rapidly, and that we're going to really see a rapid increase in the use of this technology in the public health and also in the clinical environment in future years. So thank you very much for the opportunity to speak, and I'll sign off now. Thank you.
Thank you again. Today's webcast will be available for on-demand viewing through May 12, 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.